let's start. I'll give an introduction. So Stan's actually in my lab at the moment, but the work is kind of presented from his time with Alex Fletcher. So it's all about like how cells go through the cell-phase transition, how do you model a cell-phase, and that's really the goal here. And how do you incorporate signals or other aspects which you want to go into these kind of cell-phase transitions.
Yeah. So I'm Stanley Strawbridge. I'm a Henry Wellcome Fellow down at the Stem Cell Institute. And the work was done in collaboration with Jenny Nichols and Alex Fletcher. So the experimental work I did with Jenny and the modeling work I did with Alex. But I put up the fun title, Pushy Guests, where we looked at how donor cells are able to impede host epiblast specification in the 8-cell stage chimeras through these two mechanisms of spatial crowding and FGF signal. So I'm only going to talk about one story today. If I can get my mouse to mouse. No. Let's try that. There we go. So a little bit about mouse development for those of you who have not seen this before. So what we're looking at are some two-cell stage cleavage mouse embryos inside their zona. And if I can play this. So we're going through another cleavage to the 4-cell stage, and then an additional cleavage to the 8-cell stage. This is when we make our first cell fate decision during the process of compaction. You can see that happening here, where the outside cells go on to become a polarized epithelium called the trophectoderm, and the inside cells remain a bipolar progenitor cell. These will then go on to make a cell. Embryonic fetus called the epiblast. So let's slow that down and look at the stats. This is just a fun video to bring you in. So the first steps we have here. So the first fate decision is we have at that 8-cell stage, we undergo compaction where the outer cells become trophectoderm, which is this extraembryonic tissue which will found the placenta. And that's driven by one of these key transcription factors called CDX2. And the inner cell mass will remain on the inside, non-polarized, and kind of established there by the expression of OCT4. The second cell fate decision that happens here is we undergo something a little bit more complicated. Instead of just an inside-outside decision, we have the feedback methods. So initially, we start to specify the ICM in what's called the salt and pepper pattern. So cells of the unspecified ICM will start to take on the identity of either the primitive endoderm, as identified by upregulation of a marker called GATA6, or upregulation of a marker called nano. So that primitive endoderm is founder of another extraembryonic tissue called the yolk sac, and the epiblast will make all the cells in the adult organism. And is also the source of embryonic stem cells. So that is the basis of one of the basic questions driving my Henry Wellcome project at the moment. The quick answer is no, but some people have feelings. My main idea is that it's basically local signaling gradients that aren't fully diffusible. So you think you have this fully diffusing FGF signal. So one of the things driving ICM specification in the first instance is expression of FGF4. And both the ICM and the epiblast express FGF4 and push the ICM towards that primitive endoderm identity. But why don't all of the cells of the ICM just become primitive endoderm, right? So there's something a bit more complex happening there than just a simple diffusion, diffusible morphism, right?
So it's actually to get a ratio. So
very much so. So that's what I'm going to go into with the modeling that we're going to do. So I've drawn this question mark here to be a bit provocative. Because Nestor Saez back in 2020 put out a nice paper where they did a laser ablation study and started shooting primitive endoderm cells in a not completely specified ICM. And when you remove epiblast cells, then the unspecified ICM cells start to lean towards the epiblast fate and vice versa. So whatever you remove, the ICM is able to sense what it's lacking and tries to compensate for that, right?
And how precise is it?
This is another point of kind of, some people say it's very consistent. The mouse line that the Hajantanakis group works with says 60-40. If you use a different mouse line, it will vary, but it's quite a robust system, and you will tend towards strain specific ratios.
It is not like CL, but a precise number map.
No. So it's a very plastic system.
Yeah.
So I mean, I can talk about playing with the mouse embryo all day. Like one of the fun things is you can take two 4-cell stage chimeras, put them together, you'll get a bigger embryo. You can take or an 8-cell, cut it in half, you'll get two mouse embryos that are smaller at the pre-implantation size. When you actually transfer those back into a surrogate, you get a mouse. So there's something a bit more complex happening later. Yeah, exactly. Something a bit more complex happening later on. Exactly. So FGF is one of these very important signals, which is driving cell fate specification, and it gets progressively restricted to the ICM, and then eventually just the epiblast. And you can actually control, you can alter the ICM composition through small molecule inhibitors and addition of FGF itself. So here is a nice kind of mid-stage blastocyst. The ICM hasn't quite segregated into the two tissues yet, but you can see that the epiblast here in red, expression nano, is not co-expressed with the GATA6 marker here in the green or in the blue. And if we add FGF to the system in very high non-physiological concentrations, we push the entire ICM compartment over to the primitive endoderm fate, and vice versa. If we use this MEK-ERK inhibitor, which blockades the FGF signal, then we push the entire compartment over to the epiblast identity. So the last little bit of information that I need to give you before we dive into some experiments, some modeling and some experiments, is this idea of chimeragenesis. So these three gentlemen spent a lot of time developing the methods for generating transgenic mice. And by doing so, what they've shown is that embryonic stem cells have the ability to re-enter normal development as demonstrated by chimeragenesis. So you can take the epiblast tissue out of a blastocyst, culture those cells indefinitely. You can manipulate them quite easily, genetically manipulate them quite easily. And then you can reintroduce those into a host blastocyst. These cells will happily incorporate back into the host embryo, as demonstrated when you transfer back into a surrogate mother and have contribution to all the tissue layers in the adult mouse. It lets us sample the 5-dimensional parameter space. And what I want to show you here is from previous data, previously published data, we're able to get some pretty decent fits, which give us compact support over the parameter space that we were sampling from. When we actually use the mean posterior parameter values to simulate the model or to solve the model numerically, we get in the white line is the numerical solution and the dots are the empirical data. On the X, we have the sum of the number of cells in the ICM compartment. So that's all 3 tissues. And then on the Y, we just have the trophectoderm. And you can see from the empirical data, it's roughly linear. There's some heteroscedasticity. I don't know what the implication of that is for a growing embryo, but it's there, it's something to think about.
So if you made a stochastic model of it with exactly the same rules for deterministic OD, that explains the variance?
It could very well do. I mean, I assume that it's buried in there somewhere. I bet if,
Of course, I think it might not be,
yeah,
but that was an interesting thing, that actually just putting some stochasticity into rates according to, you know, just directly turning it into probability from rates, and you might find that wrong explaining the variance.
the other thing, the other nicer thing that might, another thing that we could do is to instead of taking the mean of the posteriors, is we actually take the sets of the distributions and then plot the individual sets and see if that explains it as well.
Just simulating sampling.
Exactly. Yeah, the simply from the posterior joint distributions. Is an embryo. Different times. So you can think in these data set, you can think embryo size, the number of cells within the embryo as a proxy for time. So if we move into the ICM, this is just to show you that the numerical solution roughly fits the data. We have this nice curve where we have an increase in the number of ICM cells. It swirls back as the epiblast and primitive endoderm start to fall out of that population and lands on the epiblast primitive endoderm plane and once the full ICM is specified. And just to reduce that a bit, I just want to show that if we look at the time course of the individual compartments, that we lose blastomeres over time, as you expect. The blastomeres should go away. You shouldn't have blastomeres in a fully developed blastocyst, shown here. And then in the ICM, if we zoom in on that the ICM will tail off at some point as well. So I hope that's convincing. So we can,
And it's 50-50.
so this one is just above that, or that's what the mean parameter value predicts for that. But the original paper that we drew the data from says 60-40, but it's kind of one of those things where error bars are involved in, you know, what do you trust? Do you trust the empirical data? Do you trust the model? Where's the middle ground there? So I hope I convinced you with a simple, you know, ODE system of coupled ODEs. We can relatively simulate the population of different tissues in the embryo relatively well. So now we're going to start moving into, so that sets the groundwork for starting to build an in silico model for a chimera, for making a chimera. But before we do that, what we want to do is perturb the tissues to understand how these donor cells are affecting the host tissues. And what we did was we used both FGF producing and cells that did not produce FGF4, donor embryonic stem cells. And what we saw there is that these donor ES cells, both the wild type and the knockout, can actually displace cells from the host epiblast. So the setup is we take the morula, it's still in the zona, and we pack that with 10 wild type or 10 knockout ES cells, and we let that culture for an additional 2 days, which takes it up to the peri-implantation stage. Fix, image, analyze. So here we have some happy embryos. In the middle is our non-injected sample. We have the epiblast marked by SOX2 here in cyan, and the primitive endoderm marked in magenta by GATA4, where the donor cells are shown in, this is just constitutively expressed to get red from the donor cells just to show who. Orange ICM with a slightly larger primitive endoderm. The interesting thing that this experiment got me thinking about was if we go to the wild or the knockout, the FGF knockout ES cells, almost the exact same thing happens to the epiblast. You have lost almost your entire host population to the donor population of ES cells. And accordingly, the primitive endoderm vanishes as well. So not only do you lose that FGF signal, but you must lose it early on. So you must displace cells from the host ICM, not just the epiblast. When we go through and quantify this, So I'm going to show you a few ugly graphs like this, but we can come back to this. Each bar, each vertical bar is an individual embryo, and then it's split by colors. So in these, the gray is donor cells, the green or the cyan is epiblast, and the magenta is primitive endoderm. But the important thing I want to point out here is that in both instances, both the null and the wild type, the host compartment is completely depleted in the epiblast. So it's interesting, right? Why is that happening? Yeah, so especially the FGF null cells. That's just a wild type ESL.
What are you transplanting to identify the genetic host?
No, I think it's like for like. So I think it's just getting to the point that you don't when you're doing transgenesis, you're only injecting like a few cells. Here we are hacking in 10 cells into 8 cells. An ESL is about that big, and a blastomere is about that big. So I mean there is a size difference happening here as well, but there is some type of displacement happening.
Could be that the host cell will be quite low because it is much bigger, right? So you have to compact it with the process that's needed, and then ESL kind of missed that process.
Yeah, ESLs have already skipped to the epiblast stage. So you are putting a preformed epiblast into an 8-cell stage morula at that point. So I don't think it's so much of an advantage kind of thing. I think it is more of a simple mechanistic thing of signals and a bit of physical forces. So we can go through and quantify that. This is just a sheet of quantification. Something I forgot to explain is that when we actually did this experiment, we had the FGF nulls had a mixed population that contributed. One population contributed really well to the embryos. That's what I'm calling 10 minus high. And then another group of those embryos did not have good contribution from the donor cells. So I've unmixed those and just to show that they have the ability to do what I'm about to say. So in the first instance, the left will be the number of the cells present in a tissue, and the right will be that fraction. So the first thing we noticed is that chimeras exhibit more cells in the epiblast. So here's the non-injected. Across the board, we have more cells in that epiblast, primarily because we put a lot of ESLs into there to begin with. The interesting thing arises when we actually look at host tissue versus donor tissue. And you can see here, both in the high contribution, the 10 FGF wild types, almost the entire host epiblast population is gone in a statistically significant way. And finally, we see a reduction of the primitive endoderm. So what this is saying to me, so we lose our host ICM population prior to the specification of the ICM. Otherwise, you might see host ICM producing FGF to actually drive primitive endoderm specification, but that is not what we're seeing. So donor cells can evict the host tissue from the epiblast. And what I wanted to know is, are they killing the cells, or are they just pushing them to a different compartment? And so I thought that it might be the case that Alex and I always talk about cornflakes. Where do the small cornflakes fall in the box of cereal? They go to the bottom. Well, if you're in a 3D sphere and you add tiny cells, the big cells, then you would think that they would wiggle and move towards the center of that and push any cells that are starting to specify in the ICM back to the outside towards the trophectoderm compartment. And that's what we see. So when we actually increase the number of cells that we see, we noticed that chimeras injected with FGF null donor cells have fewer primitive endoderm and more trophectoderm. So very similar experiment, but now we have increased the amount of donor cells that we have to really kind of break the system, to push it to its most extreme limits. So right off the bat, what you see is on the 15 cell on the left and the 15 cell on the right, regardless of wild type versus null donor cell, these are just quite large embryos. And we see kind of a gradient as you move from 15 to 10 to non-injected embryos. Similarly, if we look at the primitive endoderm here, there is no primitive endoderm in the null injected, while we have a decreasing level. So initially in the previous one, I showed you that there was an increase in primitive endoderm, and now once we kind of go beyond the break point of the system, we start to see a decrease again. Here's another one of these immensely complex bar charts, but what I really want to show you here is that both at the embryo population, we have larger embryos on both sides and larger ICMs as a whole. So even mammals hatch out of an egg. So this is a hatching embryo. So for those of you who don't know, we all have a proteinaceous egg, and eventually the embryo will start to pop out of that, and there will be a small protrusion that will start, and then eventually it'll start to blow up on one side while the other side is still blown up on the inside. And you'll get a little pinch bottleneck, which is what you're seeing, these little snowmen. And we fix those and we wind up with lovely little snowmen embryos.
It is representative
It's a bit arbitrary. It's just a random sampling. Some hatch, some don't. We didn't really dive into that. Some of them we catch. I mean, it's more of a technical relic. Like, when did we actually catch them? Did we catch them, you know, a half hour too late, or it's relatively quick. So this is the getting into the quantification. So we see again the epiblast in these embryos have greater numbers in the across the board in the injected embryos, and again we see a reduction in the number of primitive endoderm cells specified in the null injected, the null FGF null donor cell injected embryos. A slight inconsistency here was we see a numerical increase in the amount of primitive endoderm like we did with the first experiment. But then, as I said, I think we have kind of broken the system once we get to the 15 cell stage, or the 15 cell injected embryos, and we actually start to deplete that tissue again. But importantly, what we see is in the null injected embryo, or the null ESL injected embryos, we see an increase in the amount of trophectoderm specified. We do see kind of a numerical increase in the 10 cell wild type, and kind of a skewing in the 15. I wish I had a better result for that, but I don't. That is the data. But it still gives me the idea that the injection of these donor cells is acting at the first fate specification. And so this led us to think, well, how can we prove this? And one way we wanted to go about doing this is to layer in the chimera aspect to our base model of embryogenesis, or base population of embryogenesis. And what we see is that our quantitative modeling suggests a role for both displacement crowding during the first fate specification and FGF signaling during the second fate specification. So here, I'm just going to run through, we're testing 3 models of chimerogenesis, where we start to layer in a few more features. So the first obvious feature that we have to include are the donor cells. So here we either have D- FGF nulls or D+ FGF wild types. In the first instance, we're just in adding in FGF induction in the sense that they contribute to this term driving ICM specification towards the primitive endoderm fate. We also allow for the net growth rate to be the same as the hosts. Slight spoiler alert, that did not work out so well. So we had to refine that model to actually include induction and a differential growth rate. So not so surprisingly, donor embryonic stem cells do not cycle at the same rate as a host embryo cell in a different cell state. It makes sense that one cell state and a different cell state do not have the same cell cycle rate. Fair enough. The final layer that we added in here, so far, the donor ES cells that do not express the FGF4 are basically inert little balls sitting in the middle of this tissue. The final thing that we added was the a spatial crowding term. So what you're not seeing here is a row, which is the original bias that we calculated through ABC over 1 minus alpha D, which is the total number of donor cells. So basically, as you increase the number of donor cells, the denominator gets, what happens? It gets very small and goes to 0. So you're basically biasing everything towards the outside, and vice versa. As you decrease the number of donor cells, you revert back to the original bias of inside versus outside. So when we actually go through and perform the second round of ABC on top of this, we're able to get relatively comparable donor cell net growth rates when we include that. And I just want to show we have compact support over the sample, the space that we sampled over. So the fun result. The gray box is the induction alone model. The slightly darker gray triangle is the induction differential growth, and the open black circles are crowding, induction, and differential growth. So when we look in the first instance at the amount of epiblast that we produce in our model, so the boxes are the data from the previous experiment that we just looked at, and we get like astronomical numbers of cells in these embryos when we do not control for that differential growth rate, which immediately told us that's wrong. Go back and fix that. When we do fix that, we see that we get a more reasonable size of the epiblast, and kind of like a nice bird bat wing kind of thing happening here. And indeed, both of these models, the induction with differential growth and the crowding induction and differential growth, fit that quite. What does not fit well is when we look at the levels of primitive endoderm. So when we look at the number of primitive endoderm, both the first and second models that do not incorporate spatial crowding here do not affect the amount of primitive endoderm being produced at the number level, which is indicative of a secondary mechanism, which we refer to as the fact that you need a spatial crowding mechanism at the first cell fate specification to actually drive the increase in the amount of trophectoderm that you see. So the hypothesis that I put forward is that host epiblast exclusion is driven by a combination of displacement crowding during the first fate specification, where donor ES cells push the host cells to the outside, and FGF induction during the second fate specification, where they are bringing in additional levels of FGF to push any surviving ICM towards, well, not surviving, any host cells that have made it to the ICM stage towards the primitive endoderm identity.
I just question about feedbacks in your equation. So, yeah, you've got feedbacks that just basically scale linearly as the more the cells. And is that really realistic? Right, that's basically saying that for any given cell, any of the other cells in the whole,
Yeah.
Thing,
No, sure.
it's impacting equally on its rates. Is that realistic? So I guess what I'm saying is you've introduced these 3 extra processes, but if you made your non-linearity you know, maybe you made them saturate.
Mhm.
Would that eliminate the need for one of these processes that you put in?
So instead of doing the linear term, we do some type of hill function
Yes, so it
instead.
would saturate. So the moment you just got the more, you know, the greater C, the more people you have.
So
So you can make that a hill function so that it's saturated to a certain level.
yeah, so I think the reason why we stayed away from that in the first instance was let's do this as small as possible.
Yeah, absolutely
Yeah.
agree. But now you're introducing at least 3 extra processes
Yeah.
that you're adding into that, right? And then it becomes a rather arbitrary thing. But well, what's your definition of simplicity? Do you want to exclude one of these 3 extra processes, but the price you pay is a more sophisticated nonlinearity, or do you keep the simple nonlinearity but introduce a different biological process? I guess what it comes down to is there actually evidence that for these 3 processes that you've introduced, there is actually a biological rationale, including evidence that they really are there, right? Other than that,
Yeah.
data.
Could you help me think of a way to show that? Because there's nothing coming to mind at the moment. I don't know if there is, and I don't know how you would go about showing that. Just
Right,
off the
well,
top of my
well,
head.
you need to have some sort of mutation where having these effects is absolutely required to even sort of qualitatively model their behavior.
Yeah, so I think in the simplest case, the hand wave argument that I would give is the fact that we've done this with the wild type and the null cells. So that is the perturbation that I'm putting forward as the mechanism. So these FGF null cells, I mean, the better controlled experiment would you would take beads of a similar size to an ESL and pop those in, hope they go to the center. But that's quite a complicated experiment. There's no
Also, this is the
Yeah,
problem,
it's a difficult thing.
because which ones you're going to add here?
Yeah.
And it becomes a little bit arbitrary. And also with your Bayesian computation, you can show that yes, you've got compact support and so on. But that doesn't necessarily mean that
It's true.
as you introduce the process, you introduce the right ones.
Yeah, sure.
It's very critical. That needs to be critical.
No. That's this is a paper that desperately needs to be published. And it's just been sitting on my desk for a little while. And I kind of wanted to present this story to see, where are the shortcomings.
These are the sort of things the review will probably come up with.
Yeah, I need to think about that because I do not have an answer for you. My answer beyond the hand wave is the simplest model moving forward.
And what my experience in reviews, what you always get is, you've got a model, but isn't there another model?
Sure. Yeah, exactly.
I can say how many times I've read that and read it. And of course, the thing is, it's not the model, it's what you do with the model, what it predicts.
Yeah.
I had this question. Then it's my fault. No. But anyway, I just heads up.
Can you preserve some of these things that can be, I mean, they can't even done a bit. They haven't done this.
We have so we went down to 5. What happened? I don't remember what happened. We did these experiments so long ago.
Still have differential growth.
Yeah.
Reduction, you might argue that's changing a little bit. To the cell and I guess that's one way you can change the and then the other differential growth is
Without affecting all of the cells at the same time. Yeah. Yeah.
Grows a bit.
might be slower growing ones.
Yeah.
I mean, I know you don't want to say this.
No, I'm thinking. No. Yeah,
Yeah, okay.
That's
exactly.
Yeah, I think what you do is you need to predict what you see.
And the 5 and then
Yeah,
That's what convinces people. It's a usual to predict it and then your experiment gives it, and then people are happy.
Extend the triangle. So
But things you need to use it predictably, right? It's doesn't quite work when you already have the experiment, unless you deliberately blind yourself.
Yeah. So the only model or the only parameters that we fixed the 5 parameters that we fit from normal embryogenesis. So this is where I say it's kind of predictive. We have fixed the parameters from the initial model during normal embryogenesis, and the ABC only predict the 2 new parameters for this model. So it is predictive in that sense,
Yeah, you're still busy.
kind of predictive. Yeah. So it's better than rerunning the ABC entirely for all the parameters, but it's not completely clean.
I mean, another thing you could do, if you now did rerun the ABC on all 5,
Mhm.
would it now predict substantially different parameters?
Oh, that's a good point. I've not done that.
That would then show that actually your model had some skill that actually reduce data set if it got it correct,
Hmm.
and it wasn't changed the condition of the data.
Yeah, no, I have not done that. That's a good idea. Okay, so this was done in collaboration with Jenny Nichols here and Alex Fletcher up in Sheffield. Jenny is now up in Edinburgh, and two very lovely students who helped me count lots and lots of embryos and take pictures of embryos, Anna Schradel and Ian Lundy Fallon. And a thanks to Kat and Nestor for really helpful feedback and kind of designing these experiments and stuff. Cool.
Maybe you should like take to like image some of the data.
Yeah, well, now I'm trying to.
Actually make the exact get calculated. If you,
It's so difficult.
Yeah.
because that's a prediction. Right?
Yeah.
Well,
If you did an experiment, what's the yes that you would be able to work out it,
yeah.
and then the answer.
So Stoyana did that. So Stoyana Alexandrova, when she was in Jenny's lab, she was looking at incorporation of single cell chimeras, where they would inject a single chimera, and then it was basically one culture condition versus a different culture condition. So 2I lift versus serum lift, a slightly weaker stabilizing thing, culture system. And they did that, and they showed that you have higher rates of death. I think she coined the term death wave in the paper, if anyone remembers that. And you see this onset of a death wave in both of them, but it's significantly higher in kind of the weaker stabilizing culture. But I think the data does exist for cell cycle rate of donor cells, which I'm only remembering now. Yeah.
Question which I have about the Houdini model, where we find the complete there are multiple parameters, and so therefore there are multiple. Are there independent experiments, more independent experiments that you can conduct to narrow down these parameters?
So, hold on, I'm just going to share my screen again. I think in pictures. Well, the data is what the data is, right? And you have to go with that.
Not with the sharing.
Okay.
Oh yeah. Yeah. So if we come back to here. Yeah. So how do we target that? So.
I mean, I'll read this.
Yeah.
You know, just go to the patients that you okay. So let's say I find the set of these 5 parameters. I guess alpha was 1, rho was 1, psi was 1, and beta was 1.
Yeah. Or do you want to go back to that? Is that easier to look at?
Yeah. And then I find a set of parameters, which is here particular kind of. I find another combination of parameters, which probably gives the same thing.
Okay.
How do I distinguish between these? Are these both plausible? I mean, in a sense, this is the same problem that I had with epidemiological models. Because,
yes.
You know, it's multidimensional optimization problem.
So the system is noisy, right? there is no one set trajectory for an embryo. They will go along and divide and specify, but still arrive at a pretty well conserved rate. So the trajectory that I'm showing you here in the white, the mean field, is an approximation of the paths by which all of the embryos take. So if they were all taking the same path, they would just sit on that manifold, right, or that line. So is there, your question, is there a set of parameters that can get you to the same answer?
Is there different combination of parameters that can get it?
So can I, not the values themselves, but the actual model?
No, the values, I guess. Let's say we don't tinker with the model. That's a
Yeah,
separate
yeah.
question.
I mean, sure, why would there should be? I mean, there's variability in the system. Or maybe I don't understand the question.
I guess what I'm trying to, let's say we've got I mean, these questions are very.
Yes.
So if I set certain parameters, set some, then I know what my final thing.
Yes.
Now, let's say I with the cell, I'm drawing an approximate that. And I know that, maybe I'll try for one row for 5, some theta 3 is probably the best fit for us.
Yeah.
Now, I could also find a path not potentially very different from this one, or it can be or can be not.
Yeah.
With a different set of parameters, alpha, the row. And then in that case, what would that mean for them?
I don't think it's, so I'm not quite sure. I mean, the thing in my head right now is that those things aren't at odds with each other because of the variable nature of the system. Like, yeah, sorry, I'm not answering question one. Yeah.
So that it's given.
Yeah.
If that gives you.
Yeah. So there is a distribution there of joint parameter sets that can give you that answer. And having that variability does not put it at odds that you can have multiple parameter sets that give you that.
Then that does not question the hypothesis, the model, the model that we have for this difference. Maybe
I think acknowledging the variability in the system is like I find to be perfectly acceptable. You should be able, in a non-determinate, we're using ODEs as a very simple approach to understand the system. It's properly a stochastic system, and that would be, in a perfect world, we would go that out. But we didn't want to spend a whole bunch of time doing billions of simulations to get to a simpler answer where we would end up with a mean field solution anyway.
And so I'm guessing what I'm trying to get at is how robust are these parameter choices?
So do I have? I don't think I actually have the posterior distributions of the error space. No, I don't have those plotted. But basically, these are quite robust in the fitting. You do get in it if you have, errors here. We're doing a sum squared error fitting. So basically, we throw the dart in 5-dimensional parameter space. We get 5 parameters out. We solve the numerical solution, and then for each numerical solution, each dot, the distance to each dot is calculated. We sum over that to get our error function, and when we look at that error function over a given value, parameter value, we get lovely minimum. And that's reproducible through multiple simulations.
Would that give an idea of the natural error?
Live data sets. What do you mean live?
Watch it.
Through time.
Like how many would that give you your variation, your balance for variation?
I think I need to be friends with Kate McDowell for that one, don't I?
don't know how many embryo.
Yeah,
Whenever you give the talk, it shows that video.
yeah, yeah. Well, I
That's
love
our page choice.
this.
Assuming she's.
I love this video. She gave me this video. Yeah, yeah, I know. So I know Veronica lives up in Manchester, can't have a gent office as far as done live tracking as well. But have they done it with the ability to actually count for the individual? There is no live triple recorder system.
You'll lose one.
Yeah.
You'll still get, I mean, but if you have two labels, presume the other one.
There was something about making. There was some issue about making the GATA6 nano recorder system. Or someone had two tissue labeling of live recorders, and they just had an absolute nightmare with it.
That could be a way to start.
Yeah.
Like if there is-
Having the live time course of the cell populations. Yeah. More data. The simple answer is more data.
That data could exist.
It could exist today.
In
Yeah.
her- It's in her slides somewhere. Just don't know how many embryos she had.
I mean, yeah, but if I'm remembering correctly, then you would really only be able to get like H2B cell numbers out. And so you would just kind of get exponential growth at some point. Which is,
I can't remember the label.
yeah, it's a bit disappointing.
I think there was a CD. She has a double label.
Did she do the Oct4 Halo in the end? Yeah, I don't. Okay.